and shadowy heart of Delphi's forgotten necropolis, archaeologists unearthed a cracked obsidian tablet inscribed with a language older than Olympus itself. Whispers say it carries the genetic blueprint of demigods, hidden instructions encoded in our very DNA. Tonight on the podcast, we'll retrace the steps from the silent tomb to the molecular laboratories where scientists race to decode the tablet's myth secrets. Are you ready to unlock the ancient code that could rewrite humanity's origin story? Greetings, friends! Greetings, cosmic voyagers! I am Kelaim Bey, welcoming you to the micro-DNA lab nested in the bowels of our stellar biome starship. As a kid, I gazed at the stars and asked, are we alone in the universe? Today, I've discovered each of our cells is its own mini-star factory. In this podcast, we'll unveil humanity's hidden universe, delving into DNA's coded cosmos. Ready? Strap in and fire up our molecular rocket! We won't settle for dusty textbook definitions. We are rocketing to molecular biology's frontiers, feeling each fact as if charting new star maps. Stay tuned until the end of our special Brainwaves coupon. Grab my comprehensive Udemy course, SAT Biology 101 Unlocking Life's Blueprint Level 1 at a discount, and receive a 3D printable molecule model at launch your endoscope exploration. Our molecular journey's first waypoint, the DNA double helix. Those spiral that mesmerize you. They are life's master blueprint, but what holds those spiral together? Holographic lab sign. Activate. First of all, nucleotides and base pairing. Each nucleotide comprises a phosphate, a sugar, and a base. Adenine A, thymine T, cytosine C, or guanine G. Adenine pairs with thymine, guanine pairs with cytosine. Think of them as alien civilizations sending sacred handshake signals. Perfect hits across cosmic distances. Why AT and GT? I mean, adenine to thymine and guanine to cytosine. How do weaker or stronger base pairs affect helix stability? Let's talking about helix stability and hydrogen bonds. Adenine and thymine pairs share 2 hydrogen bonds. Guanine to cytosine pairs share 3 bonds. Regions rich in guanine to cytosine boast extra stability, like reinforced starship hull segments braving cosmic storms. Close your eyes and envision the trillions of helixes in your body, each safeguarding your life's code. How do they endure the molecular void? Universal genetic language. Let's talking about universal genetic language. All life on Earth, from bacteria to humans, uses nearly the same genetic alphabet. A bacterium on one world and a plant on another speak same letters. This hits at life's deep, interconnected web. When we hear self-replicating machine, we imagine sci-fi wonders. Yet DNA perfected this eons ago. Let's replicate it. Listen to the cell division symphony. First of all, unzipping the helix. Helicase enzymes unzip the DNA like a molecular zipper, akin to opening a ship's airlock. Helicase unwinds 50 to 100 base pairs per second. Imagine reading 100 letters per second mid-flight. Oh my God. I need to drink coffee, Voyagers. RNA primers launch. DNA polymerase needs a primer. Short RNA fragments ignite synthesis like rocket ignition sparks. Polymerase's precision. DNA polymerase reads the template 5 to 3, adding matching nucleotides at approximately 60 per second. A human cell must copy 3 billion base pairs before division. That's billions of letters shorted every hour. Oh my God. Okazaki fragments and the lagging strands. The lagging strands synthesized in Okazaki fragments joined by DNA ligase. Each fragment is a precise puzzle piece snapped into place. Next, the genetic code, triplet codons, translate into amino acids. 64 codons encoding 20 amino acids orchestrate protein diversity. First of all, the codon table. AUG starts translation with methionine. UUU specifies phenylalanine. Picture cosmic data rockets roaring amino acids shipments. The second is start and stop signal. So important, so significant. UAA, UAG, UGA, halt translation like an emergency docking command. Mistakes here can cascade into molecular catastrophes. Translation mechanism. Ribosomes read mRNA, tRNAs deliver amino acids like robotic arms, and on assembly line forging living machines. and a single point mutation. One letter change can rewrite entire molecular novels, sometimes unleashing disease or profound adaptation. Intrigued? Imagine an equatorial space station colonized by aliens needing custom DNA, designing plants and microbes thriving in microgravity. CRISPR emerged, first of all. Biotechnology frontiers, gene editing, biofuel producing microbes, next-gen vaccines, molecular wizardry reshapes industry. Ethical horizons. Could we become a genetic elite? Transition from Homo sapiens to Homo technologicus? These questions leave deeper marks than any star chart. Our journey into DNA's hidden universe ends here, especially this topic ends here. I hope you have felt the power of unrivaling life's code. But our adventure countries use code brainwaves. On Udemy's SAT Biology 101, Unlocking Life's Blueprint Level 1, grab your discount and 3D model gift. Dive deeper and join me next time for Protein Factory's Ribosome Wonders. Before talking, let's drink a coffee, Voyagers.